Alright, welcome back everybody. This is the Principles of the Innate and Adaptive Immunity, Chapter 1.7 on the Spleen Anatomy. Last time we talked about the lymph nodes. This time we'll be talking about the spleen, and the next time we'll be talking about the Peyer's patches. So make sure to follow along and smash that subscribe button, and also the like button if you like this. And also let me know in the comments below what you would like to see in the following lectures, and also anything you would like me to clarify. So this is epitope immunobiology. Let's begin. Alright, so the spleen is the fist-sized organ that's situated essentially right behind the stomach. It really has no direct connection with the lymphatic system, so make sure you know this. This is no direct connection with the lymphatic system. Last time we talked about the lymph nodes. They do connect the lymphatic system with afferent and efferent lymph vessels. This one has no connection to the lymph system and really is only connected to the blood. Okay, it collects antigens from the blood and is involved in immune responses to blood-borne pathogens. All right. In addition to that, it also has a secondary function, or it could actually just be a primary function, but it also disposes of senescent red blood cells, okay, in the red pulp. And we'll talk about what that is, but essentially one of the other functions of the spleen is not just to participate in B cell and T cell maturation, but rather also in just the cleaning up of red blood cells. Senescence just means old. Okay, and they contain something called periarteriolar lymphoid sheath. Okay, it's called PALS. It's this blue stuff right here surrounding a blood vessel. Okay, and these PALS contain mostly the white blood cells. Okay, so this is where white blood cells are. This is where the T cells are. The lymphocytes surrounding the arterioles running through the spleen form isolated areas of white pulp. So you don't see it here, but essentially there'll be a lot of lymphocytes that essentially collect into one spot, and then they form something called white pulp. And I'll show you that in the next diagram. And we also have something called marginal zones around the follicles. So these would be the follicles. You got marginal zones here. Is rich in macrophages and dendritic cells, and they also contain these marginal zone B cells. And I'll essentially tell you about that too. So, but those B cells are right here, and down here is macrophages and dendritic cells. Okay, let's put M and D. And the follicles located along the PALS contain mostly B cells. So this is right here. These follicles contain mostly B cells. All right. So let's go through the anatomy of a spleen here. I basically exploded that image that I showed you in the previous slide, but this time there's a few extra things and extra labels. So this is essentially a large version of what we see in here. Okay, except I didn't draw the blood vessels. Let's say there's a blood vessel here going through here, or even through here like that. Okay. Or just essentially they're in these areas. All right. And this is the red pulp. So this is the spleen in the below, and there's a red pulp on the outside. So this would be everything around here. That's the blue color is actually the red pulp. Okay, that's why it's labeled just on the outside here. And what happens is that there's these blood vessels that go through, and all these blood vessels that's run through can also drop off red blood cells. Okay, senescent old red blood cells in these regions and also these regions. It doesn't really matter where, but these old red blood cells I mentioned earlier will be cleaned up. Okay, so there will be no more. Okay, they're just essentially old red blood cells that don't really function properly anymore. In terms of the lymphoid system, so the blood goes through here, and the naive T cells, or even or actually let's start with the naive T cells. They can actually then enter into these areas right here. Okay. These are the PALS, okay, or the periarteriolar lymphoid sheath, okay, these areas here, right here, okay. And when they enter out here, they can essentially sit around and wait for other antigen-presenting cells to come by. But essentially, when does one come by? Okay, so it goes through here and it exits. An antigen-presenting cell goes through here, enters into the PALS, or even goes through here and enters into the marginal zones. So I mentioned earlier that there's macrophages and dendritic cells here. These macrophages and dendritic cells essentially are the antigen-presenting cells, right? So they present these antigens on their MHC complexes on the cellular surface in the PALS and also in the marginal zones to activate T cells. So the naive T cells will be then differentiating into effector T cells in this region right here. Okay, in for example here or anywhere along the blue areas. Okay, and also a little bit in the marginal zones, but barely so. And so they get activated into the effector T cells. Again, those are like helper T cells, regulatory T cells, memory T cells, and also cytotoxic T cells. Okay, and then once those ones are activated to become effector T cells, these antigen presenting cells can also interact with not antigen presenting cells. The antigens can also interact with the B cells that are in this region right here. Okay, this region is called not the marginal zone, the B cell corona. You see here I labeled this B cell corona. This is where all the B cells are. It's called B cells. And these naive B cells could then now be activated into an effector B cell. Again, that's the plasma cell. And of course, that requires the help of a T cell, a helper T cell. Okay, so the helper T cells that are differentiated in these areas, okay, can then migrate to the B cell corona. Okay, and here they actually participate in the activation of B cells. Okay, and these B cells that eventually then can perform clonal expansion will perform this in the germinal center, so this yellow area. And then they eventually continue to grow into the occasional memory B cell as well, and also the plasma cells. Okay, and of course, at this time, then they can start to secrete antibodies, and other effector T lymphocytes then can migrate into the blood, go through the blood, and of course exit the spleen. So this is only a little portion of the spleen, but essentially this would go out of the spleen, way far out of here, and then go back to the areas of infection on the tissue, the peripheral tissues, and so on. So really, this is how the spleens essentially are areas where T cells and B cells can mature, in a slightly different way compared to lymph nodes. But in this case, it's only to the blood-borne pathogens, and it's because it's not connected to the lymphatic system. This one is connected to the blood, not the lymphatic system. All right. And so just a quick summary of what we just went through here, and how the spleen contributes to the lymphatic system. Or not lymphatic, sorry, your lymphatic immunology. The red pulp, okay, is the area where there's a disposing of red blood cells. The lymphocytes around the arterioles in the spleen form isolated areas of white pulp. Okay, oh, I actually forgot to mention that earlier. It's right here. This is the white pulp. The white pulp can then form areas where a lot of the white blood cells are, and so on. So again, the blood vessels that can just be running through here, then a lot of them can aggregate into one area and make this white pulp. And what we have here is that the PALs, the periarterial arteriolar lymphoid sheath, okay, surrounding the arterioles contain all the T cells. They can participate in the activation of B cells as well once they form helper T cells, because they migrate from there to the marginal B zones. Okay, and the marginal B zones can also have B cells that essentially reside there. Then these B cells, as they continue to proliferate, can form the germinal centers. Okay, so the marginal zone contains a lot of marginal zone B cells and macrophages that were filtered from the blood. I mentioned that earlier. And of course, upon activation of T cells by antigen presenting cells in the PALs and marginal sites, the T cells migrate to the germinal center to help activate B cells. And again, germinal centers contain many intensely proliferating B cells in the plasma cells. And of course, then this mounts an adaptive immune response. Okay, so this participates to the adaptive immunity. Okay, just like lymph nodes does. And this again is an area where the adaptive and innate immunity are linked through antigen presenting cells such as macrophages and dendritic cells. And lastly, again to mention, this takes 6 to 9 days to occur for these B cells and T cells to be activated by these antigen presenting cells in the spleen. And so that again explains why when you're sick, it's because your innate immunity couldn't fight off the infection. But in this case, the adaptive immunity takes about 6 to 9 days to fully proliferate and become a mature army to then detect these antigens. Of course, then after going through the spleen, they can exit the spleen through the blood, go back to the areas of infected tissues directed by chemokines, so that they can then fight the infection. And then you typically feel better after about a week and a half. And if you don't, you really should see a doctor. All right, great. So this summarizes, and so this finishes the section on splenocytes and the spleen. Next time we'll be talking about on chapter 1.8 the pairs patches and how this area contributes to the lymphatics, your immune system. So again, I draw on a diagram here. Of course, click here to get to the next video. And make sure to subscribe and follow along because there's a lot of interesting lectures going along. Hit that like button as many times as you want. And of course, let me know in the comments below if there's anything that you'd like me to cover for following lectures or even clarify from these lectures. This is Epitope Immunobiology, and we'll see you next time.